We can score. We avoid dangerous situations. We are faster than our opponents. Or we recognize someone in a split second in a crowded street. Our brain constantly reacts to the current situation around us. But some brain processes do not have such a strong connection with things happening at one specific moment. Feelings that emerge, a slowly growing desire, or changes to your body that unfold gradually. We feel euphoric after a great achievement, or satisfied after a good meal. The development from child to adult is also a gradual process directed by our brain. Such changes can be observed throughout the animal kingdom, for example in the caterpillar that transforms into a butterfly. How does our brain regulate all these gradual changes? To answer that question, we have to take a journey inside our brain. Our brain consists of billions of nerve cells that form a complex network by connecting to each other. A typical nerve cell consists of a cell body and different protruding branches that can send or receive signals. Most branches receive signals. They are called dendrites. A nerve cell also has one protrusion that sends signals to others, called the axon. Signaling takes place at the specialized junctions between axons and dendrites of nerve cells called synapses. These are real nerve cells seen through a microscope. The dendrites are colored with a green dye, the axons are red, and the synapses are shown in blue. Nerve cells form a complex network. They communicate by sending fast chemical signals at the synapses. These signals are mediated by chemicals called neurotransmitters. But the more gradual changes that our brain regulates start with the secretion of other chemical signals, small proteins called neuropeptides. Neuropeptides are produced in the cell body and transported through the different branches of a nerve cell in small packages called dense core vesicles. Under a microscope, transport of neuropeptides can be observed in live nerve cells. The neuropeptides are colored with a red dye, and you can see the small vesicles moving as they transport the neuropeptides. The vesicles are transported through all the different branches of the nerve cell. Sometimes they change their direction or they stop for a few moments. They will move through the cell until they receive a signal to release their content, the neuropeptides. Dense core vesicles release neuropeptides by fusing with the plasma membrane, the outer surface of the cell. The neuropeptides are now free to move through our brain and the rest of the body. When they come into contact with another cell, this cell receives a signal to do or change something. In this way, neuropeptides regulate many processes in our brain and body. The short green light flashes are the neuropeptides being released in live nerve cells. We see the exact moment the vesicle fuses with the plasma membrane. How neuropeptides work exactly is the subject of our research. We want to know how the vesicles are transported and what determines the direction they travel in. What is the molecular mechanism that triggers their fusion with the cell membrane to release the neuropeptides and transmit a neurochemical signal in the brain? If neuropeptide communication is dysregulated, severe problems may arise in the brain and the rest of the body. For example, eating disorders such as anorexia or obesity, developmental and growth problems, psychiatric disorders, or disrupted sleep patterns. By studying neuropeptide communication, we hope to find solutions for these problems. And thanks to the neuropeptides secreted in your brain during the last few minutes, you can now leave this movie feeling great.